Hello everyone, I'm Sandeep. I'm the Senior Global IVD Product Director for FEPON Biotech. Today webinar is about vitamin D. I'll be speaking about a novel sandwich immunoassay that FEPON has developed for 25-hydroxy vitamin D. But firstly, I'll tell you about FEPON. FEPON came into existence in 2001. We are one of the top high-quality IVD raw material and one-step solution provider. When we came into existence in 2001, our focus was on the raw materials. We were developing the antibodies, antigens, and the enzymes for most of the IVD industry. So these are the critical raw material for the IVD industry. From 2009 onwards, we started focusing on the IVD applications. So we started producing several of the reagent solutions for the multifarious assay formats, and we became one of the main suppliers of the raw materials for the emerging markets. From 2017 onwards, we started focusing on the one-step solutions, where apart from the IVD raw materials and the reagents, we were also venturing inside the open instruments. So we started developing the open analyzers such as for CLIA. From 2021 onwards, we became one of the major cooperation providers for the IVD industry. We started focusing on the ecosystem cooperation. So we forged most of the partnerships and the strategic cooperations with many of the IVD industries. And our vision for the future is we see that continuous innovation as well as focusing more and more on the emerging technology that is very important for any IVD industry. So that is our focus. We will continuously evolve, innovate, and focus on the new products and the technologies. We are already 23 years into the existence, have got more than 1400 matured products. Further, we have numerous patents for the various technologies and the products that we have created. FEPON has more than 2500 business partners throughout the world. So in this particular webinar, we will be discussing about vitamin D. We will be discussing about the sandwich immunoassay that we have developed using FEPON's critical raw materials. And I'll also share with you our vision for the future. Firstly, let's discuss about vitamin D. As all of you know, vitamin D is a small molecule of around 400 Dalton. Within the human body, it circulates bound to vitamin D binding protein as well as bound to the albumin. Further, it is also available as a free form. 25-hydroxy vitamin D is one of the major biomarker for many diseases. That is why its prevalence as well as the detection is very, very important. So total 25-hydroxy vitamin D, it comprises of 25-hydroxy vitamin D3 as well as 25-hydroxy vitamin D2. But D2 is only prevalent in a very small fraction, and that too only if it is present in a particular subject. The half-life of vitamin D3 is around 1 day, while vitamin D2 is around 2 days. Let's discuss about vitamin D metabolism. As all of you know, sunlight is a major source of vitamin D. So 80% of the vitamin D is coming from the sunlight. Around 20% comes from the diet. So depending on whether it is sunlight or the dietary source, ultimately the different forms, they goes like to the liver, where with the help of the enzyme 25-hydroxylase, it converts the pre-existing forms of vitamin D into 25-hydroxy vitamin D. And 25-hydroxy vitamin D is the main biomarker for the vitamin D status. So that is why the detection of 25-hydroxy vitamin D is very, very important. And that is what we are focusing in this particular webinar. So 25 hydroxy vitamin D further metabolizes in the kidney to 125 hydroxy vitamin D, which is also a useful biomarker for kidney and the parathyroid diseases. Now, what are the conditions where vitamin D determination is important? Most of us, we already know that it is one of the major bone metabolism biomarker. So, in case of the parathyroid, kidney diseases, as well as the intestinal disorders, the determination of vitamin D is very, very important. Then there are conditions like osteoporosis, osteopenia, or the skeletal disorders, where its determination is also very critical. Further, there are certain states or the determinants in the patients, like if they are obese, overweight, or they have certain disorders such as for the kidney, or they might be of advanced stage, that is more than 50 years old, and they have like low exposure like to the sunlight, or depending on the color of the skin, so it is more prevalent like in the dark skin. So then there are certain subjects which are also not consuming that much of the dietary sources that contain vitamin D. In all these states, the determination of vitamin D is very, very important. So that is why it is one of the major biomarkers for the bone metabolism, for kidney, and for the several of the disorders and the disease states in humans. Now let's discuss about the levels of the total 25 hydroxy vitamin D. As you know, the optimum level that is prevalent in humans is between 19 to 50 nanograms per ml. So these are the optimum levels of the 25 hydroxy vitamin D. But if your level is between 10 to 19 nanogram per ml, then you have a mild deficiency. This could be related like to the risk of osteoporosis or secondary hyperparathyroidism. But you have severe vitamin D deficiency when the levels are below 10 nanogram per ml. So in which case it could be associated to osteomalacia or rickets. So then the determination of 25 hydroxy vitamin D becomes very critical. But there are also certain states when your vitamin D levels are higher than 50 nanogram per ml. So if your levels are between 50 to 80 nanogram per ml, then you have increased risk of hypercalciuria. But if your levels are higher than 80 nanogram per ml, so this is prevalent like in several of the kidney failure patients where the levels are very, very high. So then you can have like the toxicity. So, but toxicity, most of the patients you will see the levels are much higher. So it is higher than 150 nanogram per ml. In this particular slide, we will be discussing about the risk groups as well as the determinants of the vitamin D. Now, there are several diseases which makes you more prone to vitamin D deficiency. So these particular risk groups are chronic diseases, like several of the patients might be having like the kidney, heart, or the liver failure, or they might be having transplant. Then there are other groups of the subjects that have gastrointestinal diseases. Then you have granuloma forming disorders. There might be patients that are admitted to the intensive care units, or there are subjects that have hyper or hypoparathyroidism. It also depends on the BMI. So obese persons, they are more prone to vitamin D deficiency. Then the color of the skin, the exposure to the sunlight, or there are certain respiratory disorders also that makes you more prone to the vitamin D deficiency. Pregnancy or the lactating women are also more prone to vitamin D deficiency. So all these conditions or the risk groups, they makes you more prone to vitamin D deficiency. Then regarding the determinants, if you are not exposed to the sunlight a lot, or if your BMI is very high, you are not taking most of the vitamin D containing foods, or you are not going too much like inside the environment or taking the sunlight, or you have decreased physical activity. So all these conditions, they decrease your vitamin D levels. On the other hand, the opposite of that, if you are more exposed to the sunlight, if you are taking more diets or the supplements, or you have increased physical activity, then your vitamin D levels are higher. So it is very important to have more sunlight exposure, more diet, and more physical activity to obviate the vitamin D deficiency. Now in the next slide you will see like that the vitamin D deficiency is a serious threat. So if you see the numbers, we have already crossed more than 1 billion persons that are vitamin D deficient worldwide. Now this is 1 in every 8 persons that are vitamin D deficient. But in the coming years, you will see by 2050, the numbers will more than double. So we will have more than 2.1 billion persons that will be vitamin D deficient. So this is 1 in every 4 persons. So that means we have already crossed like the pandemic levels in terms of the vitamin D deficiency, and there is a serious need why it needs to be determined and why it is one of the most used tests in the IVD community. Now more than 50% of the population is already below the optimal levels. So these are not completely deficient, but it is vitamin D insufficient. So, but most of it is a serious threat like in Asia, in Middle East, and in the Northern Europe. Now, if you see like the vitamin D deficiency, we all know like vitamin D is one of the major biomarkers for the bone metabolism. So you have the skeletal effects. So in terms of the skeletal effects, the vitamin D deficiency makes you more prone to fragility fractures. So you have more fractures in your bones. Then you have, if you are severely deficient, then you are more prone to osteomalacia and rickets. But if you have vitamin D deficiency, it makes you more prone to osteopenia and osteoporosis. Now, apart from the skeletal effects, you also have extraskeletal effects. Like if you have decreased levels of the vitamin D, it makes you more prone to autoimmune and infectious disease. You are more prone like to cardiovascular as well as the respiratory disease. Then it makes you more prone like to obesity as well as to the diabetes. Further, it has also been seen like the incidence is particularly high for cancer also in such patients. Then the muscle strength as well as the risk like for the bone functions are particularly high. So you have also impaired muscle where the function and the strength of the muscle is also impacted. But if you see the effect, the quality of life is decreased, the mortality becomes higher, and you are more prone like to acute diseases such as COVID-19 severity or the long COVID. So both the skeletal and the extraskeletal effects, they contribute to many of the healthcare conditions. Now, if you see, there are various vitamin D detection methods that have evolved during the last decades for the detection of 25-hydroxy vitamin D. So let's discuss about all of these methods. The first one is the radio immunoassay. As most of you know, it is a very highly sensitive method. It is simple, accurate, and cost-effective. But the problem is it incorporates many of the manual steps. So there is radioactive waste, and additionally there is cross-reactivity with vitamin D metabolites. So yeah, currently it is not being used a lot. Then in the second phase, most of the testing was done via ELISA. ELISA is simple as well as it is cost effective. But the problem is, again, it incorporates most of the manual steps. It takes a lot of time, so the turnaround time is a few hours, and it is affected by the sample matrix. And further, it has the same limitations of RIA, that there is cross reactivity with the vitamin D metabolites. Now the next method is HPLC. In the HPLC, the advantage is it can detect multiple compound. So it is accurate, and the advantage is also it is automated or semi-automated. But the disadvantage is that it involves like high cost. So there is low sample throughput, and the sample preparation is cumbersome, and it is also affected like by the interferences, and you require very highly skilled analyst. So all these disadvantages makes it not so attractive like in the clinical settings, where you need to analyze so many samples in a particular time frame. Now the next method that came out was point of care method, which is based on the fluorescent immunoassay. So this is fast, easy, much cheaper, and you can perform it at the point of need. So it became one of the major methods in the developing nations where the testing need to be performed at the point of need. But it has its limitations, that it has limited sensitivity, accuracy is an issue, and it is prone to the interferences. But it also has certain limitations, that it has limited sensitivity and accuracy, and it cannot distinguish between D2 and D3. I'll say like one of the major factors is like the accuracy of the test that requires more intensive efforts. Then the next method that came into existence was latex immunoturbidimetry. So it is fast, it is cheap, it is easy to perform, but the disadvantage is it has limited sensitivity, and the accuracy is also not very high. It is prone to the interferences, but it also requires a very stringent control over the temperature, because the assay is impacted a lot by the temperature changes. But during the years, as most of you have seen, LCMS has evolved one of the major detection methods for the determination of the 25-hydroxyvitamin D. In fact, it is the gold standard for the entire industry, because you can detect multiple compounds and the multiple metabolites of vitamin D. So it has very high sensitivity, it has high specificity, it is automated or semi-automated, and the interferences are separated from the matrix, so it is not affected by the matrix. But the disadvantage is the sample throughput is limited. But the disadvantage is it requires demanding sample preparation prior to analysis. So the cost of the instrument is high, so analysis is also high, because you require a highly experienced as well as skilled analyst. But it is possible in the near future, the automation as well as improvement in the LCMS can make it one of the major tests in the clinical diagnostics. But in terms of the clinical diagnostics, where we know like the speed of the testing as well as the accuracy and sensitivity of the testing is very, very important. In all those criteria, the chemiluminescent immunoassay is one of the major formats, because you can run it using the total lab automation in a fully automated way, and you can use it like on hundreds or thousands of samples in a particular central lab. So it is fast, it is simple, it is fully automated, and very, very high throughput. Yeah, it has certain disadvantages that it is affected by vitamin D binding proteins. It also have cross reactivity with the certain vitamin D metabolites. So the industry also has to devote more efforts to make it more improved in the coming years. Now, in the next part, we will be discussing about the sandwich immunoassay that we have developed at FAPON using the FAPON critical raw materials. But first, before we discuss about the sandwich immunoassay for vitamin D, we have to understand why for small molecules such as vitamin D, there is a need for sandwich immunoassay. As most of us know, for the small molecules, there are not too many options available. So for many decades, the only assay format that was in the market was competitive assays for small molecules like vitamin D, 43, T4, estradiol. So all these molecules, the competitive immunoassay has been used to date. And that is the format that you will see in many of the FDA approved kits. But during the recent years, sandwich immunoassay has also been developed for the small molecules, and it has strategic advantages with respect to the competitive. It has higher sensitivity, higher specificity, the linear range is higher, and the cross reactivity to the immunological derivatives is low. So that is one of the major advantages of the sandwich immunoassay. And the correlation to the gold standard, that is LCMS, is very, very high. But if you see at the assay format, you can predict like these are the particular advantages of the sandwich immunoassay. Because in case of the competitive immunoassay, your analytical sensitivity and specificity, it depends on the affinity as well as the specificity of only a single antibody. That is why it is limited in terms of the affinity constant. And you will find like the competitive immunoassay, it exhibits a lot of cross-reactivity to the immunological derivatives. While in case of the sandwich immunoassay, your target is recognized by two different antibodies. So this dual recognition, it increases your sensitivity and specificity of the assay and increases the range of the assay. And that is one of the reasons you will see like it has decreased cross-reactivity for the immunological derivatives. So this improves like the overall bioanalytical performance of the assay. Now, when FAPON developed like the sandwich immunoassay, we understood like what are the advantages of the sandwich immunoassay for the small molecules. And then it becomes like one of our major targets for the small molecules that we will be focusing on in the near future also. So our assay, the first step is like there is a capture antibody bound to the magnetic beads. So it binds to the vitamin D in the patient samples after the dissociation of vitamin D from the binding proteins. So in the first case, like the capture antibody binds to the vitamin D and forms like the immune complex. Then this particular immune complex is recognized by the detection antibody. Now the detection antibody binds to the immune complex, not to the single vitamin D. So that is the strategic difference for the small molecules. So when the recognition happens of the detection antibody to the immune complex, it gives rise to the sandwich immunoassay complex. Now at FAPON, we have the capture and the detection antibody for CLIA, but we are also providing for multifarious assay format. Like we can also provide you for the fluorescent immunoassay. This is one of the core assay strategies for the small molecules. and we see the strategic advantages for 25-hydroxy vitamin D. Now, in the next slide, we will discuss about the analytical performance of our sandwich immunoassay for 25-hydroxy vitamin D. As you can see, our sandwich immunoassay, it has very high correlation with the gold standard, that is LC-MS, and it has very high correlation with the predicate kit, which is from Roche. If we compare ourselves with all the vitamin D sandwich immunoassays in the market, FAPON has the lowest limit of blank, limit of detection, and the limit of quantification, which is 0.5, 1 and 1.5 nanogram per ml. And when you see in terms of the detection range, the linear range of our assay ranges from 1.5 to 150 nanogram per ml, which means we can cover the entire pathophysiological range of vitamin D. So this broad linear range is a strategic advantage also for our sandwich immunoassay, and we can detect both the isoforms. So we can detect 25-hydroxy D3 as well as D2. And our assay, it is unaffected by the potential interfering substances. We have very high stability, and the accuracy of the test is also very high. And more important is we are using our own critical raw materials. So at FAPON, we have a wide range of raw materials for different assay formats, and we can provide it in bulk because, as you know, FAPON is high throughput IVD raw material manufactured. We are ISO 13485 certified. So as per your demand, we can provide you the particular raw materials up to kilogram level. So that is one of the strategic advantages of associating with FAPON. And because we have been in the industry for the past more than 2 decades, we have multiple generations of the technology, like the single B cell technology, phase display. So we have several generations of the technologies in which how we can improve the sensitivity and specificity of our raw material and make it more and more improved with the several generations of our raw material. Now, this is the comparative analysis of our assay format. As I was mentioning, the LOB, LOD and LOQ, we have the lowest in the entire industry. And we have very good correlation with the LC-MS and with our predicate Roche CLIA kit. So most of our results are, you can say, with the limited number of the samples. But as our number of the samples will increase, we will have critically improved assay results further, which we will be sharing in the next 1 or 2 months. So, but we expect a very high sensitivity, specificity, as well as correlation with the predicate. But from our preliminary results, you can already see that we have achieved very high performance using our sandwich immunoassay and the critical raw materials developed internally by FAPON. Now, what will be our next steps for the future? As we have developed the sandwich immunoassay and the critical raw material for the small molecules. So, yeah, as you can imagine, our first priority will be developing the same formats for the other small molecules and the challenging targets. Our first target are definitely T3, T4 and the estradiol. But we have a pipeline of the projects for the small molecules that we will be focusing on in this particular area. And if you're also working on other small molecules or the challenging targets, please let us know. We will be more than happy to cooperate and work together with you. Thank you, everyone. So if you want to interact with us personally, please feel free to visit us at Medica. We will be exhibiting in Hall 3 at our booth A81. If you want to know more about our products and the IVD solutions, please send me a mail at sandeep.vashisht@fapon.com. Thank you everyone. Have a nice day. Bye-bye.